How the COVID-19 vaccine works. The virus that causes COVID-19 is known as SARS-CoV-2, and that stands for Severe Acute Respiratory Syndrome Coronavirus. It's called COVID-19 because the outbreak started in 2019. Now, to understand how the COVID-19 vaccine works, we need to know a little bit about how vaccines work in general. Now, I've recorded another video explaining how vaccines work in more detail. I'll post a link to that at the end of this video. But a quick crash course: vaccines contain either weakened, dead, or parts of pathogens. Pathogens are things like bacteria or viruses that can cause us disease. When these things are injected into our body, it trains our immune system to know what to respond to should we encounter these pathogens in the future. Now, as part of our normal immune reaction, dendritic cells, which are found throughout the body under the skin and near muscle tissue, if they sense a pathogen, they gobble it up and break it into pieces, and they display these pieces on their surfaces, and these are known as antigens. They take these things to the lymph nodes, and it helps teach our antibodies what to attack should we encounter these things again in the future. Quick way to remember this is antibodies attack antigens found on pathogens. Now, the COVID-19 vaccine is different somewhat in that instead of weakened, dead, or parts of pathogens, it delivers instructions to dendritic cells for how to make an antigen known as a spike protein, and that spike protein is what's found on the surface of the COVID-19 virus. COVID-19 spike proteins are essential as it's what it uses to get into our cells, and the virus doesn't have the option of changing it, which makes it a good target for our immune response, and it's easy for our immune system to recognize and attack it. Now, COVID-19's genetic material is RNA, ribonucleic acid. Our genetic material is DNA, deoxyribonucleic acid. However, we do use RNA, specifically mRNA, when we make proteins, and this is part of what's known as the central dogma of molecular biology. DNA is transcribed into messenger RNA in the cell nucleus, and then mRNA is translated into proteins out in the cell cytoplasm, and this occurs on ribosomes. And so a quick way to remember this is DNA makes RNA, and RNA makes proteins. Now, the COVID-19 vaccine takes advantage of that fact, and it contains a little mRNA snippet that codes for the spike protein. Now, if we injected mRNA directly into our bodies, our immune system would probably recognize it as foreign and attack and degrade it. So to help the mRNA get into our dendritic cells, the mRNA is encapsulated in tiny fat globules. This allows it to get into our dendritic cells. Once inside our dendritic cells, the fat globule is dissolved and the mRNA is shuttled over to the ribosome, where it then makes a spike protein. The mRNA is degraded and the spike protein is displayed on the surface of the dendritic cell. So the dendritic cells with their new COVID-19 spike proteins make their way to the lymph nodes, where they teach the immune system, okay, if you see this spike protein, attack it, and specifically make antibodies that can bind to the spike protein. Now that our antibodies know how to recognize the COVID-19 spike protein, if we're subsequently infected by COVID-19, they can help our T cells and our B cells, which are part of our immune system, recognize and destroy the virus before it gets to levels high enough to make us sick or kill us. Now, some important points. The COVID-2 mRNA only needs to get into the cell cytoplasm, not the cell nucleus where our DNA is. And even if some did get into our cell nucleus, our cells can't convert mRNA into DNA, so it's not going to alter our DNA. The vaccine mRNA can't replicate itself. Our cells had no capacity to do that. And mRNA transcripts are produced and degraded all the time by our cells as part of the protein building process. You might be wondering, why not use traditional vaccine approaches? Well, it can take many years to figure out what the ideal microenvironment is that preserves the potency of a vaccine's antigen and do subsequent clinical trials. The mRNA vaccine approach is much quicker. Scientists have been researching mRNA vaccines for over 20 years and have long thought that it was a promising way to make vaccines. Now, if you want to get deeper into it, here are a few articles that can serve as a jumping off point. Now, our mission at Smarter in a Minute is very simple: education. So it would help us out a lot if you could please like this video and please subscribe to our channel. Thank you so much.